How's everyone doing? Okay. Who thinks that we should start off our class with like a montage of Monday music videos?
Yeah.
I think it would be really fun actually. None of my kids wanted to get up and leave the house this morning, and I was like, I hear you. And so we listened to Monday on the way to school. But there's so many great Monday songs, I feel like we need to compile them all and just listen to them on our way home. Okay. Molecular biology, right? Okay. So you'll get your exams by Friday. What? Those of you who took it on Monday feel like it's been a long time. Those of you guys who took it on Friday, it was just on Friday. So that's what happens. I'm sorry. I just got some back from the testing center just now. Okay, so far, what have we been talking about? We've been doing cell cycle, and I like to start with an introduction of the cell cycle from a cell biology perspective, so you can use it as the backdrop for understanding DNA replication and mitosis. So we talked about CDKs and how they're regulated by cyclins, by phosphorylation, by CKIs. We talked about the ubiquitin proteasome system and how it's not just a trash can for misfolded proteins. There's a lot of specificity to the proteins that are degraded and when they're degraded and how they're degraded to turn off different processes. We gave some examples on how CDKs regulate the cell cycle. Basically, what do? They phosphorylate things, and that results in degradation of proteins, release of inhibitors, activation of ubiquitin ligases. A whole lot of things happen as a result of phosphorylation. So I'll just do a quick introduction, quick thing about checkpoints, and then we're going to move forward into S phase. So just keep in mind that the cell cycle shouldn't move forward when something's wrong, and that's why we have these checkpoints. And I introduced them a little bit earlier. There's your G1 checkpoint, so you shouldn't go into S phase, right? If things aren't right in the environment. If there's DNA damage, something like that. There's the S phase checkpoint. There's the G2M checkpoint, the spindle checkpoint. You don't want your chromosomes not properly attached to your mitotic spindle before you segregate them. So this is an experiment that was done. It was used to identify Rad9, which is a checkpoint-induced DNA repair protein that's induced in response to DNA damage. And so this was done in yeast, where they did, again, so much of this was done in yeast, where they had wild type and they had some sort of mutant. And they either irradiated the cells, or they either irradiated the cells, or they didn't, or they did it before and after. And they looked at the effect on cell cycle progression. Because the whole thing is, if you're going to have DNA damage, you want to halt cell cycle and not move forward. And this was looking at the function of Rad9 in cell cycle progression. So here's your wild type yeast before irradiation. Here's your Rad9 before irradiation. Looks exactly the same, right? Because it's before irradiation, there's no DNA damage. But then you've got irradiation, and then they look again 10 hours later, and you've got wild type, and you've got Rad9. And so I want you guys to talk to your friends for a few minutes. We're going to start out this Monday morning. We're looking at an experiment, since you guys are getting really good at looking at complementation assays and yeast mutant kinds of experiments. I want you guys to look at this and tell me, what does this data tell us about the role of Rad9 and the response to DNA damage? Now it's going to be counterintuitive because in DNA damage you do not want cell division to happen, right, with DNA damage, right? You want to halt the cell cycle. I want you to think about this. So take a few minutes to wake up your brains and talk to your friends, and then I want you to let me know what's going on here. Here you've got a certain number of cells, here you have more, here you've got a certain number of cells, here you have more, here's no irradiation, here's after irradiation. Discuss. You want to think about numbers. Okay, it's getting quiet. So I'm going to give you guys a hint because this had on first look that they don't look much different on first look, right? But there's some major differences between wild type and Rad9 after irradiation. You might want to look at the amount of cells per cluster, the size of cells per cluster. Okay. So someone's going to give me an answer. So what do you guys think Rad9 might be doing? Kelsey Shiflett. Something involving cell cycle arrest, right? Because you can see over here that there's some that have divided quite a bit, but then there's a lot of these that are just kind of big and stuck at the 2 or 4 cell stage, whereas here they all have this uniform expansion going on. And so a checkpoint is supposed to halt the cell cycle when there's DNA damage. And we've irradiated these cells and waited, and when you've mutated that checkpoint gene, you have perfectly normal cell division because the checkpoint's not being triggered. When you have not mutated in the wild type cells where you have Rad9, the checkpoint is triggered, and you have some cell cycle arrest. Any questions about this?
Yeah. Is it bottom?
Nope.
It bottom
I don't read today.
So I think the confusion is it's named Rad9, but Rad9 is actually not
Rad9 is, yes, this is a Rad9 mutant.
So it's not
okay.
active. Rad9 is not active.
There's no Rad9 in these cells.
Oh.
Yeah.
That always makes sense.
Yeah. So this is like an expansion of what we've been doing with talking about yeast and mutants in yeast. Yeah. Yes.
So it's probably resting in that stage because it's not allowing them to grow and they're also not dividing. So it's not getting G2 or
So it's probably, so what the comment I just had is probably arresting at an S-phase checkpoint because it's not allowing them to grow or divide. Maybe. So what I want to know is what's going on like between here and here. What do you think the difference is here or here and here? Like these are very clearly huge and not any more than 2 cells. Whereas these, there's a whole bunch. Just hypotheses.
Does those have less DNA damage?
Could they have less DNA damage? Maybe. I mean, there's no answer. It's just a discussion. I mean, I guess there's an answer. We'd have to do experiments to figure it out. Yes?
Could it just be that after only 10 hours of radiation, the radiation hasn't affected all of the cells and caused enough damage?
Right. So after, and this is 10 hours after radiation. It's not 10 hours of radiation.
Oh.
You do a quick burst, and then you're letting the cells grow to see if the checkpoint's activated. Yeah, so maybe not all of them got the same amount of damage. Yep.
Do we know what type of cells they are?
They're yeast.
Well, can some the ones in clusters be undergoing asexual and ones and 4 clusters undergoing sexual?
Okay, so is there a difference in the type of cell division occurring in these yeast? I think they're the same kind. But I think that, I don't know. That wasn't in the figure. But I still think DNA damage should activate a similar checkpoint. Right. Any other thoughts? No? Okay. What do I think? I think, yeah, maybe you're not getting equal DNA damage. Maybe these cells started out as larger clusters to begin with. You know, over here you might have 1, and then you radiate, and that one might stop there or get to 2, and that's it. Depending on where you are in the cell cycle when these cells get irradiated. So things to think about. Okay, is everyone feeling awake now that you had to use your brain? It's like yoga for the brain. Okay, are we good? I see discussion over there. Do you guys have questions, or are you just?
If you go back, the cells haven't moved from where they were in the first slide to the second slide. So what you see on the middle one starts with 4 and it goes to 1.
Oh, so you're thinking if it's the same exact picture 10 hours later.
They are. They haven't moved at all.
You know what? You have a really good point, right? Because so here to here, right? Here to here. So it's almost like each one is going through like one round, like finishing the round they're in, and then it's triggering. Yeah. Very cool. Good observation.
There's like a really big cluster next to G alpha and G.
Yeah. Well, good job, guys. Good. Okay. So we talk about checkpoints. It's what you're told in school. There's these checkpoints, and they stop things. So you have to think about, well, what are they doing, right? So they actually are activating. Well, so you'll have your damage, right? And then there'll be some kind of sensor that will sense the damage. I think I got a question earlier on in the semester about how was DNA damage sensed. So this kind of starts to talk about it. We're not going to talk too much about DNA damage. That was a little teaser for later. You know, you get you guys all excited about DNA damage. A sensor is going to bind to the damage. What kind of sensors have we talked about already with DNA damage when we were talking about histones? H2AX, that's one. That's an example of a kind of sensor. And then there'll be a transducer. So some kind of, these are just classes of proteins. A protein, not classes, but categories of proteins. So these would be kinases or proteases that would recognize something sitting on the damaged DNA. And then they would do things like phosphorylate or cleave off things to activate repair proteins, so a bunch of effectors. Those effectors would be repair proteins, proteins that induce cell cycle arrest, right? Proteins involved in the synthesis of DNTPs. Why would you need to synthesize DNTPs when there's damage? in your DNA. Veto. Veto?
Veto.
Okay. Yeah, that's fine. Nope. Trying to not always pick the same people. Brian? Brian McGee? Yep.
I don't know.
Do you wanna call a friend?
Sure.
Why would you wanna make DNTPs? Why would you wanna make DNTPs if there's damaged DNA? I see 2 people raising their hands, but we'll let you call a friend for a second.
Yeah, no one knows.
No one knows what a DNTP is? I am so sorry. Too much time in school. See, this is what happens when you're in school forever, is you think, like, I'll be stirring my coffee, and I'll be like, I'm waiting for the sugar to go into solution. And my husband's like, what is wrong with you? Well, it needs to go into solution. He's like, no, it's called putting sugar in your coffee and stirring it. DNTPs are your nucleotides. Nucleotides.
So, if there's damaged DNA, they need to replace the nucleotide.
Great! If you're going to repair your DNA, sorry for holding out on the important information and answering the question. During an exam, if this happens, you should feel free to come up and ask me to clarify. Absolutely, if you're going to repair DNA, you're going to need to make DNTPs. Did you want to add to that? That's what you're going to say. Awesome. Okay. So all these effectors, they prevent M-cycle CDK activation, right? Because you don't move towards M phase. They prevent CDC25 activation. That's one of these cofactors that's required for activating APC. Another thing required for M phase. Okay. So checkpoint activation will activate repair pathways or induce apoptosis if repair is not an option. More on DNA damage repair later. Much more. It's going to be really exciting. But now that you're excited about it, you're going to, when I bring it up again, you're going to be like, yes, finally. Okay, so let's talk about S phase. I'm putting this figure up first because I got a whole bunch of panicked emails this weekend about it. So with S phase, origins are primed for DNA replication as early as late M, early G1. Can you believe that? Is that shocking? So this pre-replicative complex starts to actually organize itself at the origin, at the end of M or into G1 phase. And it's just sitting there, just hanging out, not activated, not anything, just waiting for S phase. And then DNA replication is actually triggered by the activity of SCDK. Okay, and SCDK, remember, it's this kinase, it phosphorylates a whole bunch of things, but the phosphorylation results in disassembly of this complex and recruitment of the pre-initiation complex, which then results in actual DNA replication occurring. So the pre-RC, this is basically proteins that come and bind to the origin and help to load the helicase onto the origin. And 2 of the proteins that are mentioned in the textbook are CDC6 and CDT1, and then MCM, which is the helicase complex, comes, and they just sit and hang out at the origin. And then SCDK activates DNA replication through disassembly of this complex. So it actually is involved in phosphorylation of CDC6, which results in its degradation, and also results in activation of, or I should say, inactivation of APC, which results in stability of geminin and inhibition of CD21. So this whole complex cannot form again until the next, until the cell cycle goes around again and you're ready to enter S phase again. And then you get phosphorylation of a whole bunch of things, which results in pre-initiation complex formation, recruitment of polymerase, and all the proteins required for DNA replication. And then DNA replication occurs through S phase, and, well, copied your chromosomes. So this is a question that freaked people out. The first step in initiating DNA replication is regulated by SCDK. How? The point of me asking this question is I was trying to get you to think about how the DNA looks throughout cell cycle and what SCDK is actually doing, and to understand that the pre-RC is formed and just kind of sitting there, and it's SCDK that activates it. So the options were SCDK triggers formation of the pre-RC. No, because that starts as early as late M phase. SCDK triggers formation of pre-initiation complex. Yep, so it's recruiting that complex to the origin. SCDK triggers chromosome segregation. Obvious, no, right? Right? Who picked that one? SCDK triggers helicase loading. Again, helicase loading occurs. That's part of their pre-RC formation. So that was that question. I just wanted to go through it to see if anyone had any questions about it. Hopefully the email I sent out clarified things. Yes?
So the PRC just waits for the SCDK to build up and then it's activated?
Yeah, once SCDK is active, then all these things happen to disassemble part of the pre-RC and then recruit the pre-initiation complex. Load on the DNA polymerase and you get.
So the initial PRC is inactive and that's the competition change to make it active?
The initial pre-RC is inactive and just waiting until SCDK is active. Yeah. Questions? Did you have a question, please?
For the next slide, which I have a question on it. That's what I thought. I thought that was the answer.
Good.
And then I thought it said something else.
Okay. I mean, it's always possible I check the wrong box and then everyone should be in that book.
Yeah, because question 3, it says that the right answer is SDK, triggers formation of the pre-initiation complex. And
Yeah.
it marked it wrong, and it formation of the pre-right.
Yeah, it's pre-initiated.
Pre-initiation.
But you said
No,
that says
I'm
pre.
just retarded.
No, we're all retarded sometimes. It happens to me. Actually, I'm trying not to use retarded anymore because my friend had a daughter with Down syndrome, and she was sitting at a coffee shop, and these two college students were like, "That's so retarded," and she's like, "It's never hurt me so much in my life." And we were all like, "But you know, like, you shouldn't talk about your daughter. Retarded means slow." But true. Now I always think about, like, there's probably a mom with a slow child. Anyway, so we should all try not to use that. Even though we all feel slow sometimes. Okay, any other questions about this? No? Okay. Was it clear as day? Is it clear as day? Questions, comments?
Oh, I just clicked the wrong answer.
Oh, that's really frustrating. That makes me angry. I do those things all the time. Okay. So this made me start thinking about, well, what is a substrate of CDK2, which is the major SCDK. And so I looked it up, and that's a lot of substrates. So I put this up here because I wanted you guys to think about these CDKs as not like a protein that's active and phosphorylates something. It actually goes wild and phosphorylates a whole lot of things in order to move the cell cycle forward. And so if you look at this, some of the things are things that are in the book, like CDC6. MCM is part of that helicase complex. But if you look through it, you can see you have another CDK in there. You've got histone H1. You've got WEE1. Does anyone remember what WEE1 is? We talked about it briefly in in terms of activation or actually inhibition of cyclin CDK activity. Yeah.
We could add
Exactly. It phosphorylates the cyclin CDK, and it's an inhibitory phosphorylation. So CDK2 actually phosphorylates WEE1, probably to turn it off when it's not needed, is my guess. Upstream binding factor, I think that's involved probably in recruitment of polymerase. Stathmin, who remembers what Stathmin is? Whoever took cell biology? Anyone? Now, Stathmin's involved in regulation of microtubules. Makes sense, maybe, a little bit. Here's another one, RB, retinoblastoma protein. RB, we just talked about RB and how you have phosphorylation of RB, which results in liberation of E2F, which results in transcription of these different cyclins and activates S phase, right? There's a lot on here. You don't need to know this all, but I just wanted you guys to think about how complicated this whole process really is, and how these kinases have to phosphorylate a lot of different proteins in order to activate or inactivate to allow the cell cycle to move forward. Just appreciate it and memorize it immediately. Okay, take out the paper. No, I'm just kidding. Didn't you always hate that when people would give pop quizzes? Right now. Is anyone going to do well? I never did well. Okay. So here are the major steps in DNA replication. You have helicase loading, primings, loading of the sliding clamp, loading of DNA polymerase. Then you've got this bidirectional fork movement and strand elongation. Remember? You guys all saw this before. And then you've got DNA ligase sealing up any gaps after removal of the primers and the lagging strand. Kind of flying through this because it's just an introduction, but these are your main components of DNA replication, right? You guys, they all look familiar to you, right? Okay. One thing I want you guys to keep in mind is that whole bidirectional fork movement. I'm just saying it because for some reason students seem to forget about it once you start looking at the actual replication level. People always forget that you've got replication going in both directions at the same time. So just keep that in mind. Okay, so DNA polymerase. So it actually looks like a right hand. Does it look like a right hand to you? Kind of like this. I think Dr. Ham has people bring in an oven mitt. Next time you're at home making dinner, take a look at your oven mitt. Think about DNA polymerase. Okay? So you've got two kind of points of activity here. You've got your active site that's right in the palm of your hand. And then you also have this site for exonuclease activity. What's exonuclease activity? Actually, I'm going to pick someone. Botania.
Right here.
Yep. Just think about what exonuclease might mean. And you can feel free to talk to a friend.
Is it a type of enzyme?
It's a type, exonuclease is a type of enzyme activity. Okay, yeah. So what kind of activity, what's the function? Yes? DNA repair, right. So it's actually, nuclease, you're right, is removal of nucleic acid, right, or cleaving or breaking of those bonds. Exonuclease is backwards. It's actually this backwards movement and removal of a base that's added incorrectly. So it's involved in the DNA repair. This is one of the major activities of DNA polymerase that results in high fidelity of DNA replication. Other polymerases do not always have this fidelity. If you think about certain viruses like HIV, that uses a reverse transcriptase that has no exonuclease activity. You have a high level of errors, which results in a lot of mutation. If you think about viruses and mutation and how they're always mutating and always evolving, partially due to the error-prone polymerase. So your incoming nucleoside triphosphate, so your incoming base comes in through here between your thumb and your fingers and is added on right in the palm to your growing template. So really what's happening here, so here's another way to look at it and much deeper. You've got your active site, so here's your palm where your active site is, and there's these different aspartic acid residues sticking out of your DNA polymerase that are coordinating these two magnesium ions right here. And what's actually happening is it's helping to hold the template and the incoming nucleotide in the correct position so that the 3' OH, say, it's actually right here, the 3' OH can then attack that phosphate right there, and then that's how you have addition of your new base onto your growing strand, and then this pyrophosphate goes away. Any questions about this? This actually also relates to another question that was over on the quiz this weekend. Okay, everyone happy? Okay, so another way to look at it. Here's a hand. This is how the hand looks in Albert's. You've got your DNA sitting in there, and you've got your incoming nucleoside triphosphate that's going to be added on right here. The way it looks, if you're looking close up, is this triphosphate's coming in, and this OH is available here for addition of the incoming base. Okay. I thought that was cool. I was just looking. I'm always looking for cool pictures because science, like sitting in a lecture, can get really boring sometimes. So there's a way to not have it be boring for a minute. Nucleophilic attack. Who remembers learning about this in Orgo or somewhere? Who was traumatized? I was traumatized. But every now and then I see it in biochemistry kind of things. Who's taking biochem? A lot of you guys. So are you finally being able to apply all you learned in Oregon? No? I heard an angry no. Who thinks that it's all a waste of their time, they'll never need to use it? Who thinks that they might end up standing here teaching someone something? Then that's when you have to dig deep and figure out, what was that? Anyways, there you go. This has nothing to do with DNA polymerase at all, but still, I thought it was nice to add. Okay, so questions, comments? I like some discussion in class. We want to talk about art. Who likes to talk about art? Okay, that's fine. We don't have to. Okay, so why are incoming nucleotides added to the growing strand 5' to 3' and not 3' to 5'? Has anyone ever thought about this before? Yeah, I actually never did until grad school when someone was like, it's clear, look at the structures. And I was like, ah. So it's okay if you look at structures and it makes you shudder, but it's actually very helpful to think about, right? So here's your DNA, this nice ladder. Hopefully this will look a little bit better on Blackboard than it's looking right now. And so you've got your incoming base, and remember, 1, 2, 3, prime OH. Remember, this OH is going to attack this phosphate on this new incoming base, and then those two phosphates are going to go away. They're a really nice leaving group, apparently. And then that's how you have the addition. But if you look at the backwards way, which is the wrong way, then you have your 3' OH. You have your 3' OH, and it's going to attack. What's it going to attack? And there's nothing to leave. So energetically, it's pretty unfavorable. Right? It doesn't work. Am I right that it doesn't work the other way? Yeah, I see nodding. Okay, thanks for thinking about it. Okay, so this was the DNA synthesized 5' to 3' because, well, that's what I was told in 201. Who checked that one? I guess that could be right. Huh? I know. This was kind of a giveaway. So I just wanted to put it up here because we just went through it. Okay. So the fidelity of DNA replication is important for maintaining normal cell functions, right? If there's no, what do I mean by fidelity? Who knows what fidelity means, other than it's where you can put your money for retirement? Who's saving for retirement now? Who thinks they don't have to worry about that? Someone, there's a few. You think you don't have to worry about it? Okay. Sorry, it's Monday morning. I'm feeling punchy. Okay. What am I talking about? Right. So, why is the fidelity of DNA replication important? Why? What happens if it doesn't happen right? What happens to the organism? It dies. It's not good. It's bad. Lots of mistakes in proteins then. Things don't work. Right, bad. Okay, so the error rate for replicative polymerase, this is the one that replicates the DNA, is about 1 in 100,000. That's actually fairly good. That's fairly error-free, not error-free, but not that bad. So DNA replication. So fidelity is maintained at 2 different phases of DNA replication, right? So one is during polymerization. So when you're adding in your base, if the wrong base comes in, it's not going to fit right, and it's not going to be added. But if it is added by mistake, then you have this exonuclease activity in your DNA polymerase. So that's 2 different ways of preventing the wrong base from being added. I thought I had a slide in here. Bummer. Oh, there it is. Okay. Okay. So, like I just said, a mismatched base pair won't fit well in the active site of DNA polymerase. So if you have a mispair, so the way it should fit is the outline here, and the way it is fitting with the wrong pairing, you can see it's just not overlapping with the proper spacing. And purine mispairs, all these mispairs, they just don't fit right. So when they bind to each other, it doesn't fit and it gets kicked out. So they have a different shape than correctly paired nucleotides. And this is 3' to 5' exonuclease activity that most DNA polymerases have, and that's what it uses to remove the final nucleotide from the DNA if it's incorrect. So here's your hand again, and you've got your active site, and you've got your base that's been added incorrectly, so somehow it made it in there despite not fitting well. And what actually happened is this mispaired end is transferred, so it's transferred down into this exonuclease site, and that's where the mispaired base is removed. And then the end is transferred back up to the polymerase active site, and where the correct base can be added, hopefully. Any questions about this? Yes?
So when it's forming on the DNA template, it's going 3 to 5' on the template, but it's forming 5 to 3'.
So you've got your, so now I'm going to get turned around. So 5' to 3', so your 3' going in, right? And then when you've got an error, it kind of moves backwards, and it's 3' to 5' to remove. Does that make sense? Okay. Any other questions? Okay, so after the addition of an incorrect base, so this is what I just said. The rate, okay, so after addition of an incorrect base, rate of incorporation of the next base is slow, allowing time for exonuclease activity. So remember, you need that strand to be pushed down into the exonuclease region, and because the incorrect base was added, the next base can't come in very quickly, and that allows time for the exonuclease activity to remove the incorrect. Yep.
The 100,000 error you said for the endpoint, but I was just thinking of the numbers, right? Not including the additional grouping enzymes.
That's
It is.
No, that's just polymerase.
Okay.
Just polymerase. Yeah. Yeah, because if you look at we're not going in. Yep. Okay. So when you talk about proofreading, you've got the incorrect base. It's either recognized by shape, so it doesn't fit well. Then the strands transfer to exonuclease activity site and it's excised, and then the strands transfer back to the polymerase active site for addition of the incoming MTP. Okay, so I always like to go back to viruses, and so if you think about HIV has its own RT, reverse transcriptase, which it uses to reverse transcribe its RNA genome to DNA, and it has a huge error rate. 1 in 2,000, quite different from 1 in 100,000, to 1 in 4,000, and that's because it has no exonuclease activity. But this is great for the virus, right? The virus is evolving at a really high rate, right? Because every single time it incorporates the wrong base into its genome when reverse transcribing, then it evolves into a newer virus, right? Pretty awesome. Questions? Thoughts? Excitement? No? I put that there to remind myself of something else. Okay, that's it then. That's all we got.